So far, we've talked about inheritance of genes that are on chromosomes that are not sex chromosomes. And so now we're going to look at sex linkage and what is the inheritance pattern of genes that are carried on the X chromosome, because they don't necessarily follow exactly the inheritance patterns of other genes are on other chromosomes, which we call autosomes. Just a reminder of the chromosome composition of sexually reproducing organisms. So we're using human beings as an example. So in human beings, we have 23 pairs of chromosomes altogether. I'm showing you the chromosome lineup of a female in here and of a male here. So the first 22 pairs of chromosomes are called autosomes. So if we were going to draw a line, these are all autosomal chromosomes. They're autosomal. Okay. We also have our sex chromosomes. So in a male, it would be an X and a Y chromosome. The male determining gene is on the Y chromosome. For a female, it's going to be 2 X chromosomes. So then when we write the inheritance patterns of genes that are on the sex chromosomes, we either remember the naming convention. For a female, we write the X chromosomes, and then we write that naming convention that we have for a gene as a superscript on the symbols. So let's say if we're looking at the gene for color blindness, normal is big C, and then color blindness is little c. So this big C stands for color. Okay, so that would be a heterozygous in a female. For a male, males only carry 1 X chromosome. The color blindness gene sits on the X chromosome. So this male only has 1 X. So it could be either big C or it could be little c. And obviously for female, you could have 2 big C's or you could have 2 little c's. You can have all these combinations. Genes that sit on the Y chromosome, well, obviously, since only males have a Y chromosome, they're only going to express that gene, show that gene. So then the superscript goes on the Y chromosome. And we don't really run into many situations where the gene is on the Y chromosome. But let's just say some gene, I'm just going to call it gene A. Then we would be writing it as a superscript on the Y chromosome. Okay. Now, the fact that these genes are on sex chromosomes, their inheritance pattern shows sex dependency. And this is what we call X linkage. So just as a review, humans have 23 pairs of homologous chromosomes. 22 pairs of them are what we call autosomes, and 2 sex chromosomes, either X or Y. XX is female, XY is male. X and Y, as you have noticed in those images, they're not exactly the same size, so they're not homologous over their entire length, but there's enough homology between X and Y that allows them to line up together on the metaphase plate. So the first incidence of sex linkage was identified by Dr. Morgan, who studied genetics in flies. And the phenotype his lab was looking at was the color of eyes. Most flies in nature have a red eye, and that happens to be the dominant allele for red eye color. And some of them are they have white eyes, so they don't have red eye color. And I want to emphasize here that just because something is dominant doesn't mean it's more abundant. So don't mistake a dominant allele for abundance or strength or whatever. All a dominant allele means is that it can be expressed both in a homozygous dominant individual and in a heterozygous individual. That's all it means. Okay, so when Dr. Morgan's lab was studying the inheritance pattern of eye color, he noticed that there is sex dependency in terms of the ratios that he observed. So it depends on which sex he was looking at. So the cross that was made was a male fly with red eyes. So obviously the male has the dominant allele, and then the female was a homozygous recessive. So the alleles that are made by this male are here, lined up here, and then alleles that are made from the female are right up here. So both alleles are the same. Now, when you cross them, two X's will give you females. So both females, not both females, all the females from this cross always had red eyes, and all the males from this cross had white eyes. So scientists noticed that when they compared the sex of the fly and the color of eye, it really depends on the sex of the fly which color eye they had. And that was when X-linked genes and the fact that some genes are inherited depending on the sex of an individual was discovered. So human sex-linked disorders affect mostly males. So why is that? Well, first of all, many sex-linked human disorders are recessive. So why do we see them in mostly males? Well, because males receive a single X-linked X chromosome. So if you think about it, let's talk about in terms of hemophilia. If we say normal hemophilia is big H, and a hemophilic patient would have the little h allele. So a female could have a number of different genotypes, right? So a female inheriting a hemophilia allele from her parents could either have this combination or it could have this combination depending on the genotype of her parents. So females have a 50% chance of getting away with getting a disease but carrying the disease gene. But a male really does not have another X chromosome to rely upon for a 50% chance of survival. So if a male inherits the recessive allele, which is disease causing, it is going to get it 100%. So that's why conditions like hemophilia and color blindness and muscular dystrophy, you see it more often in males than you do females because of just the way the sex chromosomes that are present in males. Females can be carriers. This is called a carrier. Females can be carriers but not be sick, but males don't have an option to be carrier. So, studying human genetics using pedigrees. Now, with flies and plants and any other or model system that geneticists use, scientists have the freedom to set up crosses as many as they want, and usually with, for example, with a pea plant or flies. Those are great model systems because the generation time is short, and then there's many babies that are made at each generation. So that allows geneticists to do statistics. It's easy to do that. But with human beings, you really can't do that. You can't set up crosses at will to study human genetics. So all scientists are left with is what they have in human families and different generations. So instead of trying to rely on statistics, scientists try to kind of make sense of inheritance pattern of diseases. So that's why they create a pedigree analysis. So in a pedigree, a square is male and a circle is a female. Now in this image, they are shading normal and healthy, let's say, because we're looking at alkaptonuria. This is a disease. It's a recessive trait. They're showing them with different colors, but mostly in most of the pedigrees that you see, the affected individuals get a shading. So this would be someone who is affected. So you shade its symbol that is related to the person who is diseased. Obviously, you don't kind of go outside the line, but I don't have a lot of control. So this one would be a female that is affected by the disease, but a normal male would get a clear symbol. So it's a normal female. Okay, so an important thing to recognize with studying pedigrees is that you cannot rely on Mendelian ratios to predict mode of inheritance. So if we give you a problem like this and we ask you whether what kind of inheritance pattern there is, you don't go take a, let's say, look at these parents and say, oh, they have a 2-to-1 ratio, it means whatever. You really can't do that because you don't have enough samples, you don't have enough data points to be able to do that kind of statistical analysis. So what you have to do is rely on the behavior of genes throughout generations. Now, one of those behaviors is that recessive traits tend to skip a generation, so you can easily identify them. So what do we mean by skipping a generation? It means a case like here. So these two parents, they do not, neither of them have the disease. But all of a sudden in their children, you will see one of the children expresses the disease. So that means that the normal, the healthy version of that gene is dominant, because you really can't get a child that is sick without the parents of the child to be carriers. So therefore, if you're looking for clues as to the type of inheritance of a particular disease, try to see if you can find instances where you see the symbol, the person who carries the disease skips a generation. So when we say skips a generation, we don't mean the entire generation. Just find parents that don't express the disease, but all of a sudden they have children that do. So again, heterozygous individuals are called carriers for recessive traits. So these individuals, this mother and father, they are carriers of the allele, but they don't express it. Genotypes cannot always be deciphered for sure for those expressing a dominant phenotype. And we saw that before when we talked about the test cross. So for example, for this individual and this individual, we really don't know exactly what their genotype is, because there could be both big A and big A little a possible. So is in here. Sometimes there's more than one possible mode of inheritance. And that's because, again, coming back to not having data. So you really, the best way to deal with problems that involve pedigrees is to go with the process of elimination. Solving pedigree problems could be a little intimidating, so I wanted to share some hints with respect to making it a little easier to solve that. So always follow a process of elimination. If you're given a problem and you're given a number of choices for the mode of inheritance for a characteristic that you're following, then always start with trying to find evidence in the pedigree that would eliminate a particular possibility. So what does that look like? For example, if one of the choices is Y-linked inheritance, meaning that the gene responsible for the property you're following is on the Y chromosome, then with a simple scanning of the pedigree, you should be able to rule it out. So if Y-linked inheritance is one of the choices, then what you should see in the pedigree is expression of that characteristics only in males. So if you see in the pedigree there are females that are showing the characteristic, then the gene responsible for the characteristic cannot be on the Y chromosome, because females don't have a Y chromosome. Another thing you want to look for is for recessive traits. Recessive traits tend to skip a generation. So what does skipping a generation mean? It means that if you can find parents who do not show a characteristic, but all of a sudden they have a child that has that characteristic, that's the hallmark of skipping a generation. So if you can see parents who don't show that characteristic that you're looking at, that you're following, disease characteristic, whatever, if the parents don't show it, but the child shows it, you can confidently say that the trait is due to a recessive allele. Always start by trying to find evidence to rule out an X or Y inheritance. Those are easier to figure out, easier to decipher. Now, I want to emphasize that if you can't find evidence to rule out a particular mode of inheritance, then it is still a possibility. So sometimes you get a pedigree in which more than one mode of inheritance is possible. Let's solve this pedigree together to apply some of those hints that I talked about. So here's a pedigree. The shaded symbol shows that that particular person is presenting with a disease. So we're following the inheritance of this disease. Squares represent males, circles represent females. So let's go through solving this problem. You're given 4 different options for the inheritance of this disease, and then we said that we're gonna start with the process of elimination. The first thing that is easiest is disorder on the Y chromosome, and we said that with disorders on Y chromosomes, the only individuals who would be expressing the disease would be males. So if in this pedigree I can find females who express the disease, then it cannot be inherited on the Y chromosome. And you can see that I have this female here, many females that show the disease. So this is not a possibility. Answer D is ruled out. Then we said recessive disorders are easy to spot if you can find parents who don't show the disease, but their children do. But we look at this pedigree and we see that we don't find any indication of that here. So I have parents here who don't show disease, but all their children have, and we don't have any other possibility of that. Does that mean that recessive is ruled out? No, it means you did not find evidence to say whether or not the inheritance is by a recessive disorder. So we have to choose another, go to the next hint. And then we said analyzing disorders on the X chromosome is easier than autosomes. Okay, so let's see if we can do that. We can find evidence to rule these out. We're going to start with a recessive disorder on X chromosome. That means any individual who has the disease, like this female, they have 2 X chromosomes, and let's say we show the symbol of disease with, just for argument purposes, A and little a. So in this case, little a would be the disease allele. So the genotype of this female would be this, little a, and this would be a male, so they would have an X chromosome and a Y chromosome. Now they're not showing the disease, that means they're carrying the dominant allele. So let's see what we can expect in their children. Now we take a look at one of the easiest way to do that is to look at the sons of this female. All of her sons should show the disease because males only have one X chromosome and they get it from their mother. They don't get X chromosome from their dad. So let's focus on their children. Yes, he does have this, and the other son also has this. So we cannot find so far any evidence against this. So let's look at the daughters. The daughters get one of their X chromosome from their mom and one from their dad. And if the disease is due to disorder on X chromosome, none of their daughters should show the disease because they inherit the dominant allele from their dad. So let's look at their daughters. They have the daughter who is not showing the disease, that's fine, but the next one does show the disease. But we know that this daughter inherited the dominant allele from her dad. So this evidence rules out disorder on the X chromosome, recessive disorder on the X chromosome. So let's look at the next generation. So let's see if the disorder is dominant on the X chromosome. So then any individual that showed the disease must carry, so there are two possibilities for this female. Must carry at least one dominant allele, and anyone who's showing the recessive trait, which is normal in this case, would have a recessive allele. Okay? So can we find in their children any evidence to rule out dominance? Let's see. Okay, so let's go and take a look at their children. In this case, we have a male child who is showing the disease, so that means they inherited a dominant allele from the mom, which is possible. This female is not showing the disease, they're normal, that means they are homozygous recessive, and they have to get a recessive allele from a mom and from dad, and which is possible if this is a genetic possibility. We're really not able at this point, we're not able to tell whether she is homozygous or heterozygous, so this is a possibility. Then we have a female here who is showing the disease, and she would have to have a dominant allele, which she can get from her mom, and then a recessive allele from her dad. Same thing in this individual. What about this female? The same thing as in here. These two are the same. Okay. This son also is similar to this. So they have a dominant allele that they get from their mom and a Y from their dad. So we really can't find any information to rule this out so far, but we're not done yet. So let's take a look at this couple. We have a female who is homozygous recessive because she's not showing the disease. So then what do we expect in here? This couple have two sons and they get their X chromosome only from their mom. Males don't get X chromosome from their dad. So with a female like this, you would expect both sons to be normal, but we see that one of them is not. This result alone allows us to remove this as a possibility. So therefore the answer is this. Okay, so I hope this shows how you can go through the process of elimination and look at the data so you can solve a pedigree problem.